I'll talk to you about the description of the microbiome and how to read a microbiome paper. I will remind you what microorganisms are. I'll tell you what the microbiota is, why it's studied and how it is studied. Importantly, because you're not here to gather general information, I will explain the significance for human health. And then I'll give you some future prospects. All living beings are divided into three major domains: bacteria, archaea, and eukarya. The terminology is important, and it's good that you learn some basic terms. So the microbiota is the list of the microorganisms present. The microbiome is a different word, and the best way to remember this is you know that you have a genome. Your genome is your set of genes. The microbiome is the set of microbial genes in a given environment. The metagenome is the experimentally determined DNA sequence from an entire microbiota community. You could talk about the microbiota of your husband or your wife. You could talk about the microbiome of your pet dog. But if you want to describe the metagenome of your pet dog, you have to extract some material. It could be cells, it could be feces, and you have to sequence the DNA. It's simply a diagram of all of the cellular types, all of which compose the microbiome. They could be visitors. They could be tourists, like probiotics, because probiotics are commonly not found in the consumer. They impart a benefit, so the consumer takes them in, but they typically do not last too long and they're gotten rid of until the next probiotic consumption. And we could also refer to vandals, which are bad guys who are breaking things. And these correspond to, for example, gut pathogens, which come in and do some damage. So the take-home message is that many, if not all, of the members of the microbiome are being tested for beneficial effects. Of course, probiotics are 20 years ahead of this research because these are microbiome members which have a known beneficial effect for over 2 decades or longer in many cases. Also, fungi and yeasts in the microbiota. Some of you may know that, for example, Saccharomyces boulardii is commonly used in Europe as a probiotic. Another component which is not well understood are the bacterial viruses in the microbiome. There is a huge diversity of viruses, of bacteria, and there's a special name for these viruses called bacteriophage. We don't really know the functional significance of bacteriophage in controlling the microbiome. There's a current hypothesis that these phages control the levels of certain bacteria. The bacteria on our bodies have co-evolved with us over thousands of years. We don't have random collections of bacteria. We have bacteria that have been selected by our bodies over thousands of years of evolution. And we have different bacteria to other animals. More importantly, if your microbiota changes, it could be a symptom of a disease state, because as I'll show you in a moment, we now know that many human diseases are associated with different microbiota states. There are supposedly up to 1 kilogram of bacteria in the gut, and there are supposedly 10 times more bacterial cells and 100 times more bacterial genes. So you are probably about 10% human and 90% microbial when it comes to cells. There are 2 kinds of analyses of bacterial communities, and they actually relate to the 2 terms microbiota and microbiome. If you want to study the microbiota in the gut of a person, you could take a biopsy. But if the person's healthy, you won't get ethical approval to do that because there's no reason to do it. So typically, you'll take a stool sample, which is not ideal, it's not perfect, it's not identical to the mucosal biopsy, but it's a good proxy. The first method, what you do is you do a PCR, a polymerase chain reaction, with primers which amplify up a ribosomal RNA gene from all bacteria. If you want to know what they can do, what you do is you go back to the original total community DNA and you sequence it all. And you see bacteria, you see viruses, and you see yeast, but it's about 97% bacterial, and it's a big DNA sequence file. So option 2 on this slide is the microbiome, and option 1 is the microbiota. About 48% of the human microbiota in this person who's Bacteroidetes, 51% phylum Firmicutes, and the other bacteria make up a relatively small proportion. In this example, in the top left, you have the microbiota of old people, and on the top right, you have the microbiota of younger people. This is from one of our earlier studies from 2011, showing that older people have more phylum Bacteroidetes in their microbiota than do younger people. Changes in the microbiota affect health. This is why it's important. This is a table of diseases, which I'm not going to read, and it includes things like inflammatory bowel disease, celiac disease, and diabetes and obesity. And for all of these diseases, changes in the microbiota have been reported. A good way to explain why bacteria in the gut are important is to look at animals which don't have any gut bacteria. And what you notice as the gut becomes larger and distended, and the cecum becomes very enlarged. It becomes so enlarged it impacts on the uterus, and it's hard to get these female mice to breed. And the text below mentions the fact that the digestive function of these mice is reduced, their immunity is altered, the gut motility is altered, and they have different responses to stress, suggesting that the bacteria normally present in the gut are involved in controlling this. And they also have to eat a lot more calories, because it transpires that bacteria in the gut are majorly responsible for converting dietary ingredients into available calories for the hosts. You cannot study human nutrition and ignore the microbiota. If you do, you are missing one of the major biotransformations which converts the ingredients you recommend into metabolites that the host, the consumer, needs. People can have different usage of diets if they have different microbiotas present. And my own research institute, the APC Microbiome Institute in Cork, has shown that gut bacteria can affect the brain and can have effect on behavior, depression, anxiety, and mood. The incidence of disease like rheumatic fever and tuberculosis is going down, but what we are seeing on the right-hand side Thank you very much.